We're jumping right into one of the most fascinating logistical puzzles happening inside you right now. Think about it. Our blood is basically a watery environment, right? But the fats and cholesterol our bodies absolutely need to survive are essentially oils. And, oil and water literally do not mix. So how exactly does our watery blood transport these oily molecules to every cell in our body without them just clumping together into a massive, deadly traffic jam?
To answer that, we're going to map out the body's incredible internal shipping network. We've got 6 stops on our route today: the lipid transport problem, anatomy of a lipoprotein, the lipoprotein fleet, processing dietary fats, internal lipid logistics, and finally, we'll wrap up with good and bad cholesterol.
Starting off with Section 1, the lipid transport problem. Let's quickly look at the physics of why oil and water refuse to mix.
The core issue here is that essential molecules, like triglycerides and cholesterol, are non-polar. In chemistry terms, they are completely hydrophobic, meaning they absolutely will not dissolve in water. If you just dump them straight into your bloodstream, they'd immediately aggregate and completely block your blood vessels. So how does the body pull off shipping them through the vast watery highway of the circulatory system?
Well, the body's ingenious workaround involves something called an amphipathic molecule. Basically, these molecules have a kind of split personality. They have a polar, water-soluble terminal group attached to a water-insoluble hydrocarbon chain. So by wrapping the non-polar lipids completely inside an amphipathic shell, the body creates a microscopic delivery vehicle that's perfectly suited to cruise right down the bloodstream.
Moving to Section 2, anatomy of a lipoprotein. Let's pop the hood and see how these specialized vehicles are actually built. It really is a brilliant 2-part design. You've got this hydrophilic, or water-loving, outer shell facing the bloodstream, which is what makes the whole particle water-soluble. And tucked safely inside that shell, it's protecting a tightly packed hydrophobic core full of the actual lipid cargo your body needs. You can kind of think of the polar shell as the hull of a cargo ship. That hull is made of a single layer of phospholipids, free cholesterol, and some very special proteins. The nonpolar core, on the inside, holds the actual valuable freight, the hydrophobic triglycerides and cholesterol esters being securely transported to your cells.
But how does this tiny cargo ship know where it's supposed to dock? That's where those special proteins in the shell, called apolipoproteins, come in. These are essentially intricate shipping labels or specialized keys. They give the particle structural stability, sure, but more importantly, they serve as ligands that physically interact with cellular receptors to lock in their destination. Plus, they act as cofactors for the enzymes that process the lipids once they finally arrive.
Let's hit Section 3, the lipoprotein fleet, because the body doesn't just use one type of truck for all this.
It actually uses an entire specialized fleet, which we classify by density. At one extreme, we have the chylomicrons. These are the massive cargo ships of the blood, heavily loaded with up to 85% triglycerides and very little protein. They are built for absolute bulk transport.
When you look at the 2 extremes of this fleet, it's pretty wild. On one end, those massive chylomicrons range from 80 to 500 nanometers and handle dietary fats from the intestines. On the exact opposite end, you have high-density lipoproteins, or HDLs. These are tiny, nimble little trucks, just 7.5 to 10 nanometers across, packed with a dense 33% protein content. And their job is completely different. They're the scavengers.
The easiest way to remember how this density stuff works is a simple rule of buoyancy. Fat floats and protein sinks. Triglyceride-rich particles like your chylomicrons and very low-density lipoproteins literally float to the top because they're packed with lightweight fats. Meanwhile, those protein-heavy HDLs, they sink right to the bottom.
On to Section 4, processing dietary fats. Let's actually trace the physical journey of these vehicles, starting right after you eat.
When you eat a meal with fat in it, your digestion breaks it down, and the intestinal cells package those dietary lipids straight into those massive cargo ships, the chylomicrons. This is the exogenous pathway. The chylomicrons get released into the lymphatic system and eventually dump right into your bloodstream to start making deliveries.
And this whole process is astonishingly fast. Once they hit the capillaries, an enzyme called lipoprotein lipase gets activated by an apolipoprotein on the ship's hull. It acts like a dock worker, rapidly extracting fatty acids so your tissues can use them for energy. Within just 4 to 5 minutes, literally the half-life of a chylomicron, the emptied-out remnants zoom back to the liver, guided by an ApoE receptor to get recycled.
Section 5, internal lipid logistics.
because what happens when you aren't actively digesting a meal?
That brings us to the endogenous pathway. Even between meals, your liver is hard at work synthesizing its very own lipids. To dispatch these internally created fats, the liver packages them into very low-density lipoproteins, or VLDLs. You can think of VLDLs as the liver's dedicated fleet of outbound delivery trucks.
As these VLDLs cruise through your capillaries, lipases once again strip away the triglycerides to feed your peripheral tissues, like muscle and fat. And as it drops off its cargo, the VLDL physically shrinks. It temporarily becomes an intermediate density particle, an IDL, before shedding even more fat to finally become an LDL, or low-density lipoprotein.
So here is a really crucial takeaway. Your body doesn't just build an LDL particle from scratch. It's literally just what is left over after a VLDL has delivered its fat. Once the other apolipoproteins drop off, it keeps one specific shipping label called ApoB100, and boom, this LDL particle is now your body's primary mode of delivering cholesterol to your tissues.
Finally, Section 6, good and bad cholesterol. Let's decode those famous terms you hear at every single medical checkup.
We are good and bad all the time, right? Well, LDL is called bad cholesterol simply because its designated job is to push cholesterol into your peripheral tissues. If there's too much floating around, it builds up. HDL is the good hero because it acts as a biological recycling truck. It actively scavenges excess cholesterol from your cell surfaces and hauls it safely back to the liver to be cleared out.
But we have to realize that LDL isn't inherently evil. It's a completely essential delivery vehicle. It only becomes an issue in excess. Under normal conditions, your cells tightly regulate how much LDL they absorb. They use specific LDL receptors on their surface to pull the cholesterol inside only when they actually need it. I absolutely love this biological rule because it just shows how smart the body is. To put it in plain English, cells will literally stop producing LDL docking stations when they are full. It's a closed-loop system. This proves that your entire internal shipping network is designed for precise, elegant balance. The cell only takes what it needs and nothing more.
So, as we wrap up this explainer, I want to leave you with this. We so often talk about cholesterol as this terrible thing to be eradicated. But knowing how sophisticated and essential your shipping network is, how chylomicrons, VLDLs, LDLs, and HDLs all work in perfect harmony to fuel you, is eliminating cholesterol really the goal? Or is it about making lifestyle choices that actually support this biological balance rather than fighting it? Definitely something to keep in mind the next time you're staring at your lab results. Thanks for exploring this with me today.